How many? I don't know. Actually, I don't. I can't. Yeah, I can't see how many people turned up. Let's see. Okay, so we are recording. Okay. So I'm gonna. Let's see. Should be. See if I can remember how we did it last time. What did you do this? I'll be activate. Oh, and then presents. Okay. May I have your attention, please? Thank you. All right. You remember me? Yeah? Miss me? No? Okay. Like a very Drake thing to say there. Anyways, I don't know if y'all like Drake or if you listened to the album before the midterm. If you did, you probably did well. Anyways, thank you for coming. I'm sorry it had to be so late in the day. It's the only time I was able to get the lecture hall. Before we start, any feedback on the course? You can say anything and it won't affect your grade. Any feedback at all? Any way we can improve? No? Wow. No one wants to say like this course, like,
do you think they could make it available like in Zoom as well? That kind of
okay, possibly. Thank you for your input. I don't think so, but yeah, it would be nice. It would be nice. It would be a nice option. Okay. The recordings are nice though. Some classes you actually don't even.
That's true.
Yeah. Anything else? Okay, speak now or yes.
Format for the final.
Yes, it will be 70 multiple choice questions. You'll have 3 hours, and there will be more emphasis on stuff after the midterm. So like you would expect maybe 3 or 4 questions on blood and stuff like that, but much more on CNS, cognition, muscle ANS, those kind of things. Yep. And like multiple choice, pick the one best answer. Very much like your midterm, just longer and more dreadful. And you have a shaky table in the gym and someone is saying, everything is subject to inspection, and you know the gist, right? Anything else? Okay. Before we start, I want to say thank you to our USCAs. This could not have been accomplished without their help, and to the staff who helped me book this place and everything. So we'll get started. We're going to start off with body fluids. I hope you remember. And please, you don't have to take pictures of this stuff. I don't know. You're probably scanning the QR code, but I will make the questions available. I'll make the slides available. So you're not missing out on anything. Sit here, pay attention, kind of try to engage in discussion. If you have a question, this is the time to ask. I have a question for you first. Has anyone seen the new Hunger Games movie?
Yes.
Yes? So like, the scene where they're like, what's the point of the Hunger Games? So I'm gonna ask you now, what's the point of this review session? Sorry for putting you on the spot. Yeah, that's a good one. But it's to get things wrong, actually. So don't feel bad if you mess up catastrophically. There'll probably be some things where you actually have no idea. You're gonna feel terrible. It's okay. You have, when's the final? The 13th, right? 12? Oof, okay, sorry. Okay, the final's on the 12th. Okay, so you have plenty of time to prepare for this. Okay, so I'll leave it to Victoria. And there you go.
Okay, guys, we're just going to start with a pretty straightforward select which ones are true. Yeah, I'll give you a minute.
Yeah. Yeah, you can go now.
Okay, get your answers in? Okay. Okay, so forever, anyone who said A, anyone want to argue why they said that? It's the correct response, by the way, if that makes you feel more confident.
Also, I have to say, we have until 8. I have exams, I have a presentation, I have a lot of stuff, so let's get it rolling, you know. I gotta go home, I gotta call my mom, you know. Come out too late, she gets worried. So, and we got 37 questions. So we gotta do everything.
Okay, yes.
Okay, so I could look possible elimination. I said that oxidized water from metabolism is not a regulatory loss. One YCF malnutrition is one indicator. PCF is not the majority of the one YCF. And male infants will have the same as female infants and have not delivered yet.
Good. But why is option A correct? Oh, do you want to go? Yeah.
Because the questions, other individuals have more body weight, the body fat versus leaner individuals have less body
Yeah.
fat, so there's more body percent.
Yeah, that is correct. Oh, yeah, okay. So both of you guys had the right idea. Basically, the reason why option A is correct is based on the amount of adipose tissue, because adipose tissue is a dry tissue. So that's the main cause of variation in body water. Yeah. Does anyone want me to go over the other statements, or are they pretty self-explanatory? Okay, moving on. Oh, sorry. There we go. Okay. Oh, okay, I didn't click show correct answer.
All wrong, sorry.
You're showing that.
Yeah. I'll do this one. Oh, wait, why are you like this?
Yeah, I think it was in adventure mode.
Okay, we'll just do it like that so that
Yeah,
they can't see the.
Yeah.
Okay.
Yeah.
Okay, is that enough time? Or okay, we're gonna stop. Okay, most of you got it. Just gonna go over the answer real quick. Okay, so option C was correct. And this question has a few parts. So first we have to calculate the total volume of water. You can do that pretty straightforward. We're given the mass of the individual and the percent of body water. So from that, we can calculate that there's 32.5 liters of water, and then you calculate the concentration of the medication. So we're given in the question 300 milligrams. You divide that by the total body water, and you get 0.0092. And then the last part of the question, you have to remember the body water trends. So we're told that the male patient has the same mass as the female patient. And since we know that they're both adults, we know that the female patient will have a lower percent of body water because after puberty, women have more adipose tissue. So since the male patient will have more water, that means that the medication will be more diluted, and then that means the concentration will be lower in the male patient. That make sense? Okay, get your answers in. So most people got this one. Let's go over the answer real quick. Okay, so the first part of the question, we need to calculate the ECF volume using the indicator dilution method. And we had a good question earlier. Someone was asking whether or not they'll tell you that inulin is an ECF indicator. And I would say like 50-50. They might be nice, but they might not. So I would say, just in case, you should memorize it. But using the V equals Q over C equation, we can calculate that the ECF volume is roughly 14 liters. And in the problem, we're told the total body water volume. So you just do simple subtraction. ICF is total body water minus ECF. Good. Okay. So that was it for me. Body fluids is probably just going to be 2 or 3 questions on the final. So Okay, we're gonna move on to blood now.
Thank you, Victoria.
All right, good evening. Thank you for coming. We're going to get right into the blood section because let's be honest, this stuff is not very important compared to what these USCAs are going to be presenting. Let's get into it. I just have 3 questions for you guys and we're going to roll through. I'm going to be a little bit faster than the body fluids just because we have so many questions later. Okay. Is it working? Good.
My pleasure. Thank you. Take care. Let me know if you do anything. Okay.
All right, I'm going to move on to the solution and don't worry, we're going to work through it together. Okay, so don't worry if you didn't have time. Remember in the exam, you have about 2.5 minutes per question, right? So you can't take too much time. Does this work? Perfect. Okay. So I'm gonna be honest with you guys. I hate that type of question, but I put it in because it's probably gonna be something like that on the exam. I don't like it because one of the things that you kind of just have to know is that the blood volume is about 7% of the mass if the person is normovolemic. And then of course, if their hematocrit is 45%, that means their plasma volume is 55%, and the rest is relatively simple math. So this is how you get to the 2.8 value. If you see a question like this and you can't remember that 7% value, don't panic. Try to make an educated guess. I can almost promise you that the choices on the exam would be sort of more spread apart. I made them kind of tricky here with the 2.3 and the 2.8. But on the exam, it would probably be a little more spread apart. So just try to make an educated guess. You can sort of remember that fact that we have about 5 liters of blood in our body, right? That's kind of like a known thing. So just try your best. Obviously 0.15 would be too low and something like 18 liters would be too big, right? So let's move on to the next question. All right, let's look at the answer. Perfect. Okay, let's look at the sort of why the other ones were incorrect, right? So firstly, of course, oxygen and carbon dioxide are mainly exchanged by passive transport, right? They're super small molecules that can easily diffuse through membranes. You'll learn this more in the winter as well, but it's the same thing at the lungs when you inspire. Those two molecules can just go right through the cell membranes, right? So it's not active transport. They don't require a transporter or something like that. Similar type of thing here. Albumin is a huge molecule. It's a protein, right? I showed a picture here. You see the albumin molecule, and here's, for example, the salicylic acid, which is actually way bigger than oxygen. So you can see right here why albumin wouldn't be able to just diffuse through that membrane. This one's a bit tricky, but exchange of fluids does not take place only at the two ends. Sort of in the middle, we say we have net zero movement, but that still means there is movement, right? It's just sort of canceled out because it's equal going in and out, right? So that's a pretty important concept. It's rare that in physiology you would see something just not moving, but we often talk about net zero movement, and that's what that means. Then, of course, related to that albumin, those plasma proteins stay in the plasma, and they, by osmosis, they pull fluid out. So that's what that colloidal osmotic pressure is. They pull fluid back into the plasma. And then this is just a silly little typo of course, those forces distribute between the plasma and the interstitial fluid, right? Not the intracellular. Okay, let's move on to question 3. Right. So remember, this question said false, right? All right, hold on. Show results. Perfect. Super good. All right, let's go through it. So which of the following statements about red blood cells is false? Always read the full question, right? Normally in these exams, they do it pretty well. They put it in caps and like they bold it or they underline it. So it's pretty hard to miss, but just make sure you read the question, right? So again, this A statement is kind of like one of those things you have to know. But if you read that, don't panic, right? The question says which one is false. You can sort of deduce, if you forget that it's 4 months, for example, you can deduce from other statements, right, which one is false. So don't panic, read all the statements, see if you can see which one is false. Their shape allows for maximal surface area for diffusion, right? These cells are super specialized. They have one function, transporting oxygen to tissues. Everything about them is optimized, as you'll see in the other statements as well, including their shape, that biconcave disc that allows for not only maximum surface area, but also lowest diffusion distance, right? To really maximize that diffusion. So then C and D, these are sort of related statements, right? So it is true they do not have any subcellular organelles. They're not like the regular cell, as you would say. Most of their sort of volume inside is made up just of hemoglobin, as I said before, super specialized for just transporting oxygen, right? And then, of course, in D, they generate energy through the use of glycolytic enzymes. Does anyone know how a cell that does have organelles generates energy? Mitochondria, exactly, right? Powerhouse of the cell. So essentially, glycolytic enzymes just means glycolysis, which is the process that happens right before all the stuff that happens at the mitochondria, right? And that's it generates less energy, but red blood cells need less energy. Anyway, you don't need to worry about that too much. But yeah, they don't generate the energy at the mitochondria, of course, because they don't have one. Right, that makes sense. And then of course, white blood cells are obviously bigger. Those are like fully developed cells with organelles, which kind of makes sense why they're bigger, right? Any questions about any of that? No. All right, so we're going to move on to the sensory section.
Up here.
Okay, hi everyone, I'm Emma. So we'll get started with the CNS sensory. Wait, Isha, how do I go next? How do I, oh, this one.
Yep.
Okay, here's the first question.
So then, once you decide, you want to call it quits, you can.
Show results.
Yeah. First, I usually like to block voting, so they can like sit in their film if they get it wrong.
Right, and then show results, and then I can swipe to the.
Yeah. So I do like block voting, show results, and then this one you can also show the correct answer with that.
Okay. Wait,
Yeah.
you sure you can't see the retinal ganglion cell? The retinal ganglion cell? There's no picture of it. Okay, wait, I need to show you guys the picture of the retinal ganglion cell because it
My
wasn't
bad.
shown there. Okay, sorry. There's the cell. The ABCD should be the same as on the polling.
That's my bad.
Okay, do we feel like we're ready? Okay. Okay, so that was the correct answer. Does somebody want to explain why light in the center and dark in the surround is the correct answer? I can. Anyone? Yeah, exactly. Does anyone have any questions about that one, or does that all make sense? You can also see this is from Dr. Cook's slides. And so that's basically what your colleague just said. Yeah, are there any questions about that one? Yeah, so you'll expect that if Dr. Cook's gonna ask any like lesion or retinal ganglion cell questions, he has to provide the diagram because otherwise you can't really do it unless it's like a lesion question where he's like describing maybe the impairment, but other than that, you should be getting a diagram. Any other questions? Okay, we'll move on to the next one then. Okay, here's the next question. Let's go. Okay, so most people said week 3, and the correct answer was week 3. Does anybody want to explain why that was the correct answer? Yeah, go ahead.
Neural explain what that would do with strengthening the neural group and then for neural two.
Yeah, so these are the slides for reference. So at week 3, we can see neural groove development, and like your colleague said, at week 4, that develops into the neural tube. I know this question might seem like very specific, but it's very possible that you could get a question that's this specific. So just be mindful of that. Okay, we can go on to the next question, unless does anyone have any questions about that? Okay. Here's the next question.
I think that because this stuff is freshman back then. Yeah. Like maybe. Yeah. That's why I wanted to like put you towards the front so that I know you have to file.
Okay, how are we feeling? So here are the results. Most people said that there is glycogen in the brain for storage of the required glucose, being the incorrect answer. And that is correct. Yeah.
Actually incorrect. On the slide, it says very little glycogen in the brain, so that implies that there is glycogen. Like all you just watch.
I think. Yeah, so there's very little glycogen. So I guess it depends on how you would interpret that. I would say that, so there is glycogen, but it's not necessarily for storage, because the brain can't really store very much glucose. And so basically the main idea that I was trying to get with this question was remembering that the brain needs like a really big supply of glucose, because unlike other tissues, for example, muscle, it can't store as much of the glucose in the form of glycogen. So you're right, on his slide, it does say there is very little, but it's not enough to like sustain the brain in any sort of way, if that makes sense. Does anyone have any questions about that one? Okay, we can move on to the next one then. Okay, here's the next question. Okay, has everyone locked in their answer? Okay, so most people said the magnitude of the action potentials, and that is correct. Can anyone explain why that is? Yeah, go ahead. Yeah, exactly. So, okay, so you can see here that there's like 3 main things that can encode stimulus intensity. And so that's the magnitude of the receptor potential, the frequency of the action potential, and the magnitude of the neurotransmitter release. The magnitude of the action potential doesn't change because it's basically an all or nothing response. So if you get that kind of question in the exam, just make sure you keep that in mind. Here's the next question. Okay, so now we can go look at the answer. And most people said, well, I guess there wasn't a huge consensus on this question. The correct answer was A, produces a sharper population code in secondary neurons. Does someone who chose this answer want to explain why that is? Yeah, go ahead. Guys, can you be a little bit quieter so you can hear? Yeah.
Like refines the signal as it approaches the CNS. So like the secondary neurons are closer to the spinal cord than primary neurons.
Yeah, exactly. So you can see kind of in this image here from Dr. Cook's slides, basically what you said, that it's going to be acting on the second order neurons as opposed to the first order neurons. And then it is producing the sharper acuity on the second order neurons. And then the interneurons are not activating the secondary neurons, they're inhibiting them, which is shown by lateral inhibition. And then it doesn't prevent the brain from narrowing the stimulus down, it actually helps it do that. And then it's not top-down processing because it's not coming from the brain. So top-down is when it comes from the brain and goes down. So this is basically the opposite. Does anyone have any questions about that one?
Well, yeah, just what does population code mean? I'm just confused about the words.
Population code?
Like in this context.
Yeah, so it basically just means like the firing of the neurons and like how that works. So basically when you have when like your brain is trying to understand where something is, for example, it's going to interpret a population code, which is just like the way that the neurons are firing.
Okay.
I think it's less likely that you're going to see language like that on the exam if Dr. Cook didn't explicitly say it that way. But that's essentially what it means.
Okay.
Any other questions? Okay, we can go on to the next question then. Okay, so now we can talk about the answer. So most people said lesion of the left dorsal column and the left interlateral column, both at the lumbar level. And that is the correct answer. Does anyone have an explanation as to why that would be? Yeah, go ahead. Pardon me? Yeah, just say it.
So first I sounded positive. Right?
Yeah, exactly. So, in case you guys had trouble hearing, basically, when you know that there is loss of touch and proprioception, you know you might be thinking dorsal column, and because this doesn't cross until later on in the brain, you know that it has to be on the side ipsilateral to whatever impairment you have. And then we have also loss of temperature and pain, and we know that that's an interlateral column, and so that's going to be contralateral to whatever the impairment is. And so that means that both are going to be on the left side in the left dorsal and interlateral columns. And then something important to note here too is they talk about the lumbar, it being at the lumbar level. And so that's just because you know that it's the leg that's losing the touch and proprioception and then temperature and pain. And so if it was maybe another part of the body, it might be another spinal segment. Does anyone have any questions about that one? Okay. And for these lesion questions, I think it's really helpful to also draw it out. So if you're having trouble with any of these, that would be my main suggestion. Okay, now we can go on to the last question. Okay. I'm going to show the results now. So most people said E, the brain uses a population code created by RFs to locate exactly where the stimulus has been applied. And that is the correct answer. Does anyone have an idea as to why that might be? Anyone? Okay, then I'll just explain it quickly. So you might remember from Dr. Cook's slides that this would be the receptive field, for example, of this neuron. And so clearly this receptive field could overlap with maybe this receptive field. So you know that that has to be true. So you know that this has to be, this one here has to be false because they do overlap. And then large receptive fields would result in less acuity, not higher acuity. And then this one's a little bit tricky because this is saying that the magnitude of the action potential does not depend on where the stimulus is applied in the RF. So that is not well, so that part, if we were thinking like the frequency, then it does depend on where it's applied, because as you can see here, depending on where it's applied in the RF, you're going to have a different response. And then this one also was wrong because it's not the magnitude of the action potential, it would be the frequency. And then here, RFs are defined by the area that results in the greatest activation of the neuron. That's not necessarily true because different parts are going to result in different magnitudes of activation. And then finally, this one, E, is the one that is true, because based on the receptive field, you can have a better idea of where it's supposed to be. Any questions about that one? That would be a little bit different. This is more just like the receptive field specifically labeled. Line is more, I'm pretty sure, knowing like what's corresponding to what. Whereas this is more just talking about how do you know where a stimulus was applied. So for example, if someone touches a certain part of your arm, how do you know it's there and not somewhere else? And that's because it's activating the receptive field of certain neurons in that specific area, for example. Does that make sense? For why C is incorrect. Yeah, so they are an area that activates that one neuron, but it's not necessarily the greatest activation, because you can see here that different parts of the receptive field, so if it's more peripheral, it's going to result in a lower activation than when it's more central. So it's not that like the whole receptive field causes the greatest activation, like different parts have different activations. Does that make sense? Yeah. Okay, awesome. Any other questions?
It says that the label by code is the brain knows the modality and location that this has happened. So if we advise the minister would say,
okay, yeah. Okay, yeah. So then, yeah, so I guess the part where it talks about the location, then that part is related to the label line code. Yeah. Yes. But not necessarily the modality, because that would be something separate. Any other questions? Okay.
Thank you. Okay, squad, we're doing nerve synapse next.
This one's to go next.
Yeah.
After like say like 25 or 30, like lock it down, lock voting, hybrid, and then you show results and then you show results. Right. So perfect. Take away.
Okay. Hi, everyone. So my name is Omar. I'm one of the USCAs for the nerve synapse section. Just before we start, quick question. How many people are in pharmacology? Okay, so we have a couple. So my questions are a bit pharmacology based, but also obviously tying into the questions that the material for nerve synapse. So a small sneak peek into next semester. Okay. Perfect. So I think we're good to go. So yeah, this is the correct answer. There's a decrease in neurotransmitter release. Does anybody have any questions or any comments on why the other ones are false or why this one is true? All straightforward. Okay. I'll just head to the slides. Well, so basically, this question is referring to this slide from Dr. Ragzo's section. As you are blocking calcium channels, you won't have the relay of neurotransmitter release. So one of the options was there would be a depolarization of the cell membrane. So there is a depolarization, but that happens prior to the calcium channels having an effect. So that's why it's just about neurotransmitter release, and it's a decrease if you're blocking the influx. Okay, next question. Okay, so I think we're good to go again. Yeah, most of you got the correct answer. It's the same level of AP generation. Does anybody have any questions or comments for this? Yeah. Action potential, sorry. Anybody else have any questions? Yeah. Yeah, so they might give it to you, but like usually in the question, it would be clear enough of the increase. So you know it's usually -70, -60, or the resting membrane potential, and threshold is usually 50 or 55, depending on the diagram. But they're going to give you like clear values of like 25, 50, something big enough that you can clearly tell, like, okay, I'm only depolarizing by 1, so I know for sure I'm not generating an action potential. But if I'm depolarizing by 30, that's like much above the threshold. Any other questions? I'll just head to the slides again. So once again, this is referring to action potentials being an all or nothing event. So once you reach the depolarization, you produce an action potential. You don't produce half an action potential by more or less of a depolarization. You just need to reach the threshold, and then you're generating a full action potential. Perfect. Next question. So these all tie into the nerve synapse section, but you may be able to tell I'm studying for FAR 300 final exam at the same time with all these drugs and mechanisms of action. Perfect. So once again, 88% got the right answer. It's blocking sodium channels. Does anybody have any questions or comments about this question? Any reason? So I can get back to, I'll get to the slides and where it describes this. So like clearly in Dr. Ragzo's slides, there's one slide in particular that talks about local anesthetics, anti-epileptic drugs, and how there's sodium channels that are blocked. And you may remember because he talked about tetrodotoxin and bacutoxin, something like that, the slide before. And what happens is basically it's a local anesthetic, so you want to make sure that there's no feeling, you want it numb, you don't want any sensation in that place, so you want to block any action potentials, any depolarization to occur, so that's why you're blocking sodium channels. Any other questions about this? Or... Okay. So my section is a bit shorter, so this is the last question for me now. Perfect. So 91% said all of the above. So quick trick to this question, if you see that two are right and you don't know about the others, just make sure that there's an all of the or all of the above. And this deals with one of the last slides in Dr. Ragsdale's section. So basically you had one slide over here about neuromodulators, and I basically took one option for like each sentence or each piece in the slide. So we know that glutamate and GABA also activate sorry, this is about serotonin. It activates metabotropic receptors. They are neuromodulators. They're not the direct fast flow of information, and they modulate global neural states. So your primary neurotransmitters are going to be your glutamate and GABA, as those are the ones that are actually causing your depolarization or main inhibition, and the others are just affecting the transmission of glutamate and GABA. Any questions on this slide or this question, or anything on the nerve synapse section that is burning questions? Okay, so if you guys have any questions on the USCA practice problems or anything else, just you can ask me at the end of the review session, or send me an email, or post on the discussion board. And then, yeah, so we're good to go. So these questions were a bit more application type questions. Dr. Ragsdale is a big fan of application type questions. The more standard pure memorization questions, you can find more in the USCA practice problem set, so make sure to do those. And then you can try these again for one of those application type questions. Great, thank you.
Okay, take your three fingers and
Okay,
you just go boom.
sick.
And then just boom, boom. See, like this is how you go back and forth.
Okay,
So
and then I just click the down button in the slides.
yeah.
Okay.
Go here. This is the first cognitive motor one.
Yes, that is mine.
So once it gets like a number, you can usually 25 if we want to keep going a bit faster. Lock the loading. show results and then show the correct answer, and then you can go back to the slides. But
Okay. Wait, so this, so I do this, then I lock this one.
yeah, I'll be here for it.
What
I'll-
if
I'll
they
show-
show correct answer and I don't lock the vote?
No, you have to,
Okay,
because we want them to sit in, misery that they got it wrong.
okay,
So they don't forget it.
okay,
Because on
cool,
the exam, you won't be able to change your answer,
fair.
right?
True, true, true,
Right?
true. Okay, is it on? Do I talk into it?
Oh, yeah, this is on, yeah.
Okay.
Yeah.
Hello?
Yeah, they can hear you.
Okay, hello. My name's Brianna. I'm one of the Cogmotor team mentors. No, USCAs, USCAs.
Yeah.
So this is the first Cogmotor question that I made for you guys. Please send in your answers and I'll lock it somehow.
Do it now, do it now.
When we get to like 30, do you think we can get to 30?
Yeah, we're gonna aim for 30.
Come on, like 2 more people.
Yeah, there
Okay.
we go.
And then I click this
And then show results.
and then this one.
And
Okay,
then show correct answer.
yeah. Nice. Okay, so yes, it is vertical acceleration. We can see why this way, no, that way. Okay. No. Okay, here. So this is one of Dr. Cook's slides, slide 112. We see in the right corner, the fascicle is responsible for vertical linear acceleration. We can also see the other vestibular organs and what they're responsible for. Any questions? No. Okay, cool. This way. One. We'll go for 30 again. No.
Oh, that's what I should ask for Christmas. A laser pointer.
Seriously?
Yeah, it's a good thing. Imagine you're just presenting. Are you a cat or dog person?
Stop biting me. I think that's allowed.
Bro, you're tripping.
No, I kid you not. Like later, after I calm down, they'll learn to like me. But like, oh, 33. Okay, we're gonna lock the voting, show results, and then this one, right? Yeah. Okay. Very good. Looking at the slide. Thank you. We see that REM sleep here is also known as paradoxical sleep because its characteristics are kind of like when you're awake and alert. We've got low amplitude and high frequencies. Yeah. Yeah. What's up?
4, 3, 2, 1, and then rep, or is it 1, 2, 3, 4, something else? And then, in this case, because this guy goes down. 1, 2, 3, 4, and then it goes back, and then it
Yeah.
goes back.
Yeah. It goes.
So we have to read 1.
Yes. Yeah. It cycles like periodically. Yeah. Okay. Oh yeah. What's up? Oh yeah, for sure. So REM sleep is also known as paradoxical sleep because it's kind of like when you're awake, even though you're actually in deep sleep and dreaming. So its characteristics in the EEG look like you're awake with the high frequency, low amplitude. Oh.
Fair question. Like, can you just put this?
Okay, yeah. So it cycles throughout the night, stage 1, stage 2, stage 3, stage 4, and then you got your deep sleep REM, and then it'll cycle again and again and again until you start coming into your wakeness. Yeah. I don't think so. It might be good to check the slide again, though. I can only remember that it like cycles like this. Yeah. Yeah. Oops. Okay. Okay, question 3, please submit your answers. We get 2 more people, please. Okay, sick. Block voting, the results. Nice, yeah. Okay, we look here. We see on slide 140 of Dr. Cook's slides, in procedural memory, short term is widely distributed throughout your brain, and then in the long term, after consolidation, it'll go to your basal nuclei, your cerebellum, and your premotor cortex, because it's responsible for like your skill behavior that you'll like be able to remember for like the rest of your life, riding a bike, walking, stuff like that. Yeah. Okay. Oh no, this way. Question 4. Okay. Block. Results. Yeah. Okay. Yeah, y'all are doing really good. So y'all are right. Language comprehension will be most affected. If we look at our slide, we'll see that language is usually in the left hemisphere. I think it's 90% left hemisphere, 10% right hemisphere. Broca's area is responsible for your articulation, and your Wernicke's area is responsible for your comprehension. So if it's messed up, you're gonna have problems with that. Okay. Cool. Okay. This way. Voting. Oops. Yeah. Okay. Very good. Okay. For question 5, we recognize this slide. This is pretty important because it's got a lot of information about your type 1A and type 2 afferents. Slide 160 of Dr. Cook's slides. We see that the 1A primary afferents are signaling dynamic changes in muscle length and some static length. They're also rapidly adapting. Okay, cool. Next question. Cool. Oh, yeah. Really good. Damn. Okay. Yeah. Okay, that's perfect. So probably no questions of this one, but just in case, we can look at our map. Your size of body structures is related to the number of neurons dedicated to their motor control, as well as the degree of skill that's required to operate that area. So larger means more neurons and higher degree of skill. Okay, last question. Okay, you guys got it? Reduced dopamine input to the basal nuclei. Looking back at the slide, we can see right there, causes reduced dopamine input to the basal nuclei. So the treatment is increasing dopamine concentrations in the brain. Are there any questions about this section overall, or any of the questions asked? No. Okay, that's great. That's all from me. Thank you for your attention.
Oh, okay. Hello, I'm Cindy. I will be taking care of the questions for the muscles and ANS questions. There are 7, I think. So let me just close for a second. Okay. We'll wait also to like 30 people or so. Oh, I can answer. Okay. That's okay. Okay. Does anyone want to say why they chose diaphragm or not? Didn't choose diaphragm first? Yeah, you can technically control if you're breathing or not, even if a lot of that is like when you're sleeping, you're not controlling that, but you can technically still control it. Okay. Then we'll skip the slide. Next. Oh, I have 5 minutes. Questions. Okay. Oh, did I not show the correct answer? Okay, I'll come back after.
Is this Brianna's one?
Okay, let's go 9 more. Okay, 1 more. Anyone? Come on. Okay, let's go. Okay, show results. Oh, Jesus Christ. Okay, at least we know rigor mortis is caused by not having enough ATP. Cool. I will show correct answer. Okay. Go to. I went back. Okay. So next slide. So if you can see in this picture here, wait, what was the other? Myosin heavily dissociates when ATP binds to it. Okay. Did I? Give me a second. Dissociates.
We have a question?
Yes. There might be, because if there's this big of a, let me check. Yeah.
I think I made the error.
What do you mean? But yeah, it is when ATP, well.
So do you mean,
Yeah,
do you mean the power stroke option?
for this one.
Just that one,
Yeah. Oh,
the first
the
one?
first one. Oh, yes, up here.
Oh, yeah, that's my
Yes,
bad.
yes.
I'm sorry.
Yes.
My bad.
There is a typo. ATP plus PI is probably not a thing. So yes, it's when ADP down here dissociates, it comes off. Here we can see a change. You guys see the cursor? Okay. You can see a change of conformation, and then also here when there's ATP hydrolysis, there's also a change in conformation. Does anyone want any explanations for the other options, or are we okay? Yes. Okay, right. So she's it's because it doesn't dissociate because of ATP binding to it. It's because after the power stroke and it comes off, it dissociates because there's a change. You'll probably learn this in Bio 201 next semester, but there's a change in the affinity with the myosin site groups. That's going to come off, and then the ATP is going to come on. Does that clarify things, or? Okay. Yeah. The power stroke, yeah, you can understand it like that. When it comes off, it gets rid of the ATP and the ATP, and then the ADP and the PI, those come off with the power stroke. And then up there was also a question. Are we okay now, or? Okay. Next. Temple for rain, okay. Also, guys, careful with the wording. This is which of the following is wrong. Right, we're almost there. Two more people? Okay, let's go. Okay, show results. Nice. Okay. Not bad. Some of them are really okay. Does anyone want to explain why hypertrophy is the first thought? Does anyone want to say why this is wrong? Yes. Exactly. Yes, sir. Okay. And then that's basically what I also put in here. Yeah, the increase in size and then the rest are all right. Just not much to say. Okay, next one. What happened with this one? Okay.
All right.
Keep it. Because you'll understand something from it. was none of the above.
That's the answer?
What?
That's the answer? Oh,
That'd be really bad.
it's not even an option.
Yeah. All right, 2 more. Okay, let's go. Oh, nice. All right. Next. Anyone wants to explain? No?
Yes?
Let's go, Jason.
Okay, I'm afraid that. And here.
I mean, you're not wrong. But if we go with Dr. Ragsdale explanations, because to fly with to flap their wings, at least chicken have to generate a lot of force, and this is what needs where do you go? I didn't write it. Oh, fast glycolytic fibers, because they don't need the myoglobin as a source of O2 because they don't use oxidative phosphorylation as a source of ATP. Whereas when you're standing for a long time, which chicken usually are doing, they need a lot of myoglobin to bring the O2 to the mitochondria to make the ATP from oxidative phosphorylation. And I'm guessing the myoglobin it's the thing that makes it more tender. Okay, next question.
What's your favorite nervous system?
What's my favorite nervous system? Parasympathetic.
You like the parasympathetic
It's
tract?
chill. Yeah.
You like to rest and digest?
Yeah.
Oh, oops, I just gave the answer to.
You're probably didn't hear, hopefully.
They definitely.
Yeah.
This is cool. You really have ample? Yeah.
No, this isn't. And even have
What's that?
it's $3. And they can
See, like I don't know why we don't use this kind of stuff. We have all this cool technology. but we
Yeah.
don't put it to use.
well, they also have a hard time. Okay, can we hit 30 for this one?
Nice.
Okay, let's go. Okay, show results. Okay, perfect, awesome. Anyone wants to explain? Quickly. No. Well, it's just.
Yeah.
Oh. Exactly. Okay.
Perfectly said.
2 more questions, let's go. And then he has a lot.
No, I just have 6,
Okay, so send me, okay.
but 3 of them are super hard.
Okay.
Well, maybe.
Hopefully.
What if they just like have somebody nail it? Oh, farm 300. It's on the 7th again?
Yes, sir.
That's off.
6:30 to 9:30. We're all grinding that. Wait, it was on the 7th, like the first day last.
6:30, 9:32.
I swear they're doing this on purpose.
You buy the NTCs? You have to,
Yeah.
which I don't like, you know?
Yeah, I know. Like they're forcing you to pay extra for stuff that's not even that official.
You know, like I don't mind because pills like they give it back, but like this could easily be avoided if like you know
Yeah.
better.
Yeah. Yeah. But what do you mean by they give it back?
Like I mean, they use the money to host events for.
Oh yeah,
for the
yeah,
students,
exactly.
You know?
That's why like whenever there's an event, I'm like, I paid for this shit.
Yep. Yep.
All right. Almost there, 3 more people. Okay. Right. Okay, not bad. Okay. Anyone wants to explain, or do we want to pull up the slides? pull up the slides. So this is the one. Okay, so I basically just edited the slide so you guys can see what is activated by which receptors are on what. Is this going to be posted after?
Yeah.
Okay, you guys can get better screenshots after. But like this, you have a good summary slide with everything on it, and you can see all the receptors, all the things that act on them. And yeah, what's wrong is what doesn't act on them. Okay, any questions? Last question. Oh, I wanted to change this one.
I like this one.
Oh, okay.
This is like a quirky question, you know? It's a good one to end it off on.
Okay, that's fair. That's fair.
What's your favorite part of the brain?
Part of the brain.
I like the thalamus.
Oh, that's fair. I mean, if anything, the one thing I thought of was the CTZ.
Of course. Farm 300.
Yeah. All right, 3 more. Let's go. Come on, guys. We're 50 people in the room. Let's go.
One more.
One more. Oh,
Okay.
let's go. Okay. Results. Nice. Okay. Anyone wants to explain? No, that is also okay. So the main reason is because the thing that deals with emotions and crying obviously is more the amygdala than the hypothalamus. The hypothalamus is the thing that takes care of homeostasis, so body temperature, maintaining everything at the normal state. So also blood pressure and everything along with that. And crying isn't a lot of homeostasis. That's basically it. Does anyone have questions?
Yes.
Yeah. Huh. I think it's more that the hypothalamus wouldn't be the one controlling the crying. Like the amygdala is to some extent controlled by the hypothalamus, but the hypothalamus would tell the amygdala things not because we're sad, but because our body needs to reach to a certain homeostatic level. Mm-hmm. Mm-hmm. Well, okay, I get where you're coming from, but the main takeaway from this is hypothalamus is dealing with the homeostatic things, and then amygdala mostly, well, in this case, crying and emotions. And because I guess if you say it like that, that's also true. Okay. All right. Green. There you go. All right. Your turn.
Yeah.
Let's go.
Thank you. Okay, squad, home stretch, 6 more questions. Let's try to work quick, okay? Maybe we can get out of here before 8. Go eat dinner. My hypothalamus is saying I'm hungry. All right. It's a wrap for results. Very nice. Okay. So here, pure water has no solute, so it wouldn't exert an osmotic pressure. Osmotic pressure, this is the formula given in the slides. 0.9% saline from the lecture. You can see the math. I'm not going to go over it right now. It comes out to 300 milliosmoles. Same thing for the blood. And then, so we're comparing potassium chloride to calcium chloride. And this comes back to your general chemistry course where you have to like consider the Van 't Hoff factor, like how many, right? So how many like molecules, like the more there is, the more pressure it's going to exert. So KCl goes into 1 mole potassium ions and 1 mole of chloride ions. So this is 2 osmoles. But calcium chloride is going to go into 1 mole of calcium 2 plus. Let's superscript that. There we go. And 2 moles of chloride ions. So that comes out to 3 osmoles. So that's why E is the correct answer. All right. Any question? No? All right. Moving on. Boom. All right. Very nice. Okay, I'm going to show you an iconic slide. You all should be very well versed with this slide. You remember this one? Yeah. Okay. So, you know, it's a lot of it's just like reasoning. Okay. It seems like everyone's got it down. Any questions? Okay. If you have questions, you can come speak to me afterwards. All right, moving on. Last transport question. All right. Done. Very nice. Aquaporins, it's kind of in the etymology. Aquapor, you know, hole for water. Anyways. Here, so diffusion through carriers is actually slower than through ion channels, not faster. Sodium isn't a penetrating molecule. The co-transporter consumes ATP indirectly as opposed to directly. And the secondary active transport, because it's active, it's against the concentration gradient, which is why it needs energy. All right. Now, time for the tricky ones. Yeah. All right, gang. So before I show you the answer, like don't stress, this probably won't be on your exam. I just wanted to spook you. All right. It's more the message from this, all right? So that's what I'm trying to get at here. So oops, here we go. Yeah, so this one's the correct answer. So this is like it's common knowledge in medicine. I'll show you the slides here. So if you have a mother that doesn't have the Rh antigen on their red blood cells, but their baby does, then during birth, you have a lot of tearing of blood vessels. And so the baby, like this blood can then go into the mother and then elicits the immune response, because these red blood cells with the Rh antigen are considered foreign. So then the mom becomes sensitized. So she starts making antibodies against this Rh factor. So then if she has another baby that is also Rh positive, then the body's gonna be like, oh no, this is foreign. And it's gonna, attack the baby. And so this causes, it's like, I forgot the medical term for it, but it causes hemolysis. Yeah, hemolysis of the baby's red blood cells. So it's very dangerous. And, you gotta watch out for that. Okay. Man, y'all are sweating. I can see the struggle. All right, that's good enough. We'll just stop it. Whoa, okay, interesting. Dang. Okay, so why did why? Who said CD4? It's okay. Okay, why? Right. So you're not wrong, but there would still be B cells. Like they just wouldn't be activated, right? Okay, and you would still have T cells, but you just wouldn't have CD4 positive T cells. So same thing goes for CD8. So the answer is RAG. Does anyone know why? Anyone know why? Yeah. Yeah, so RAG is the protein essentially responsible for arranging the VDJ segments. So if you don't do this rearrangement, then you get the protein you actually make is like really, really huge and messed up in all kinds of ways. So here, this is an example of what happens. So this is flow cytometry. This is just a way of like getting an idea of like what kind of cells you have. You can stain them for specific markers. So in this case, we have CD3 and CD4. This basically, if you have both CD3 and CD4 in this upper right quadrant here, that's a helper T cell. It's a CD4 positive T cell. You see, when you get rid of RAG, when you knock it out, there's nothing. Okay? Same thing. So IgM and B220. If you have a cell that has both of those, that's a B cell. Okay? And so when you have RAG knocked out, nothing. Okay? And so when it comes to like clearing infections, you see, even if you have like an intact innate immune system, if you don't have this adaptive arm in this green line right here, if you have a RAG knockout mouse, you're not able to clear the infection. So it's just a bit of scientific lore for you. Okay, but the main takeaway here is that RAG is crucial to T and B cell development through the receptors. Okay. Any questions? Yes. Yeah, CD4. Yeah. So CD4 it's a CD stands for cluster of differentiation. It's a cell surface protein on CD4 positive T cells. That's basically how they're identified. They assist in the binding of like the peptide, the TCR with like the MHC2. They're just like involved in that whole conglomeration. Any more questions? Yes. Right. Okay. So if I were to have a mouse, you can genetically engineer mice, right? If I got rid of CD4, that would only get rid of like CD4 positive T cells. But so the question stem was asking what mutation or the removal of which gene would get rid of both B cells and T cells, right? So I would still have CD8 positive T cells. I would still have B cells. So that's why that option's wrong. Okay. Anything else? Okay, last question. This one's a doozy, okay? Oh, the beta didn't go in there. Anyways, it's okay. So wherever there's so the space here, that's supposed to be beta, and the space here, that's supposed to be a beta. All right, I'm gonna wait for it to go up to 10, and then we're just gonna, okay. Oh, maybe 15. Oh, maybe 20. Let's push it. Okay. 20, oh, 24. Maybe 25. Yeah, there we go. Whoa. Oh, okay. Okay. Very, very reasonable answer. However, incorrect. So the correct answer is this one. So you will not get a question like this. This is like a graduate level question. You know, this is what they might teach you this in med school. But the idea here, there's a key idea that you learned in class. It's the idea of co-reception. You remember co-reception? What's like the main molecule involved in co-reception? There's something special prize to whoever says it. Like 2 letters and 2 numbers. CD 28, okay, right? So that one's on the T cell. Which one's on the antigen presenting cell? What's it called? It's B7. So it's either B7, it's also called CD80, CD86, okay? It's on your slides, don't worry about it. But so what I'm trying to get at here is that it doesn't matter if your T cell recognizes something as foreign and the receptor recognizes the antigen perfectly to be a match made in heaven. If there's no co-reception, then it won't get activated. So I'm going to show you a diagram here. Okay. Let's see if we can zoom in. So in my body and in your body, we do have, unfortunately, T cells that do recognize our own proteins. Okay. So the reason that they're not activated and the reason that we don't have autoimmune conditions is because we're not in an inflammatory state. Like the body homeostatically, it's not saying like, oh no, we're in danger, activate T cells. So in the case that macrophages usually they clean up places. You have like alveolar macrophages that like grab like a bunch of junk in your lungs and like clean it up. You can also have that in your gut too. And so say it like digests one of your healthy proteins and then it presents it on MHC2 by accident because a macrophage is a antigen presenting cell. And then say by chance that you have a T cell that by chance recognizes it. Because it doesn't have this co-stimulation or the co-reception, you won't mount an immune response. So you're safe. Okay, it's called anergy. In scenario 2, however, if you have a bacterial protein, bacteria have PAMPs that bind to PRRs that then cause the induction of this expression of the co-stimulatory molecules on the antigen presenting cell. So when a PAMP binds a PRR, there's like a signaling cascade. It leads to transcription and translation of CD28, or sorry, B7 on the dendritic cell, which then here, now you have the B7 CD28 interaction. And so now this T cell is going to get activated and it's going to differentiate and proliferate. So now scenario 3, it is an unfortunate reality. And sometimes it does happen. Sometimes you catch a virus and you get type 1 diabetes, or it's been linked to Epstein-Barr virus and multiple sclerosis. It's really tragic. But what happens is the viral infection or the bacterial infection actually causes an inflammatory state. And so then if the antigen presenting cell by chance presents your self-antigen, your self-protein to a T cell that recognizes it, then it can mount an immune response. But you need to think the probability of this is very low, but given that there are 7-8 billion people on the earth, it does happen, unfortunately. Okay, so name of the game is co-reception. Anyways, with 6 minutes to spare, thank you for coming. And you being here is a big step towards doing well on the exam. Okay, we all got this. Study hard. Okay. Yes. I'm just gonna stop this share real quick so make sure I can get the recording.